Peace and blessings. Welcome back to another video. In the previous video, we have already discussed about the process of replication. And here today, we are going to discuss about the process of transcription. So we are well aware about the central dogma of molecular biology. That is, we have a double-stranded DNA, and from the DNA, we have the synthesis of RNA via a process called as the transcription. And from the RNA, we have the synthesis of the protein by the process of translation. So the flow of information from the DNA to RNA and from RNA to protein, that is the central dogma of the molecular biology. And today we'll be talking about this step, the synthesis of RNA from the DNA. Now let's take this as our DNA molecule which need to be transcribed. Now in order to transcribe the information, first and foremost thing is that we need to separate the two strands of the DNA, similar to what we saw earlier in case of replication. So the enzyme, it has to come and it has to have the helicase activity which will unwind the two strands of the DNA, and it will start transcribing using one of the DNA strands as template. So there are two important things which we should consider here if we compare this process of transcription with that of the replication. So first thing is that here in case of transcription, we do not require the primer. And similarly, if you remember, in case of the replication, one of the important prerequisites in order to start the replication, that was the presence of primer. Similarly, the second thing is that when we are talking about activity of the RNA polymerase, it has been seen that they are extremely highly processive. So they have high efficiency, they have high processivity. Although we will see that the polymerases, they initially synthesize few nucleotides, so we call, and then they basically pull off from the DNA, so we call those as the abortive transcripts. But once it manages to synthesize more or further than the 10 nucleotides, then it goes on and it completes the synthesis of the RNA transcript. So that is the second point, that it is extremely processive. Now let's understand the process of transcription. So we will take one after another. We will first discuss about the prokaryotes, and then afterwards we'll discuss the process of transcription in the eukaryotes. So here, as you see, starting from here, we will understand this. So you can see this is a double helical DNA structure. And to this DNA structure, we have a polymerase. This is an RNA polymerase, which will catalyze the process of transcription. But what dictates the binding of this polymerase to the region of the DNA? So that is basically guided by a factor called sigma factor. Okay. So now the sigma factor, it helps or it aids this polymerase to bind in a particular region we call as the promoter region, so that it will initiate the process of transcription. And another important thing to remember is that if we have a RNA polymerase which is made up of different subunits, like we have alpha, beta, omega, and gamma, so this RNA polymerase without the sigma factor, we call that as an holoenzyme, and with the sigma factor we call that as the core enzyme. Okay, so first and foremost, after the sigma factor, it recognizes the promoter region, it forms a closed promoter complex. Here you can see, followed by the formation of an open complex. So as the polymerase, it begins its helicase activity, it unwinds the two strands of the DNA, and followed by the synthesis of the short strands of the RNA, which I said earlier. These are the abortive initiations, the short RNA oligos which are synthesized. Usually it's, some books say that it's 2 to 8, some say that it's 2 to 10 nucleotides. But once it manages to synthesize more than 2 to 10 nucleotides in one stretch, it goes on to synthesize in one go, it synthesizes the long transcript, followed by the promoter clearance and the release of sigma factor. And now this sigma factor, which is released from here, it will again associate, and the cycle, it will again continue. Okay, so the next step is, again, so after the release of the sigma factor and after the promoter clearance, the next step is the elongation followed by termination. And termination, that is done by that is basically dictated by the sequence which is present in the DNA. So we have a GC rich sequence, and this sequence, once it is transcribed, it forms a hairpin structure. So the RNA polymerase, once it encounters this structure, it will then halt the transcription. So it will terminate the transcription. So this is how the process of transcription works in case of the prokaryotes. Now, although we say that when we are talking about the machinery in the eukaryotes, it has to be complex. It has to involve many other proteins. Similarly, we see here, so, so this here represents the DNA molecule. Okay. So this site is the start of transcription, and we have the promoter region. We know that the Tata box, which is present at -10 region. We say that the TATA box, that also called as the Pribnow box. Now, in this case, there are again two important things when we are considering the transcription process in case of prokaryotes and in case of the eukaryotes. So the first thing, if you remember, that during the process of transcription, the RNA polymerase, it didn't need any other additional factors. But here you will see there are n number of factors which it needs in order to start the transcription. These are called as the general transcription factors. So the factors which you see here, like the transcription factor second D, transcription factor second B, and E and H, all these factors, they are basically aiding the polymerase in order to bind this region of the promoter so that it can initiate the process of transcription. So that was one of the major differentiating points between the prokaryotic transcription and the eukaryotic transcription. So, or in other words, we can say the function which is done by sigma factor in prokaryotes, that is done by the transcription factors in the eukaryotes. So now see here. So first and foremost, we have a transcription factor second D. It comes and it binds to this promoter region, followed by the binding of some other factors. So it serves as a binding platform, and it will allow the binding of another transcription factor, that is the B, then followed by the binding of H, followed by binding of E, and multiple other factors. Now see here, so this is the polymerase, and this polymerase, it is recruited after the binding of the factor B. So you can see here, all of these factors, they have aided in the recruitment of this polymerase here. And then there is another property, that is this C-terminal tail, which you can see behind this blue region. So this tail, it also has very important role to play in the promoter clearance, which we saw earlier, as one of the important steps which helps the process to go into the elongation phase. So once this tail is phosphorylated, and this phosphorylation, it's done by one of the important factors, that is a transcription factor second H. It has also other factors. It also has certain other functions. Like this H, as I said, one of the important things is to separate the DNA, and that melting of the two strands of DNA, that's done by the factor transcription factor second H. We will have a look at the functions of these transcription factors in the tabular form ahead. So this was basically how the transcription proceeds. Now, as I mentioned, so there are two important things to remember. As I said before, in the prokaryotes, we have a sigma factor. In the eukaryotes, we have multiple transcription factors like D, A, B, F, E, H. In case of prokaryotes, we have one RNA polymerase that catalyzes transcription. But in case of eukaryotes, we have three different polymerases, which catalyze the process of transcription. So for the genes like 5.8S, 18S, 28S rRNA, we have the RNA polymerase first. Similarly, we have RNA polymerase second for catalyzing the synthesis of the miRNAs, saRNAs, SNORNAs, LNCRNAs, and snRNAs. And similarly, we have the RNA polymerase third for catalyzing transcription of tRNA genes, 5S RNA genes, and some of the snRNA genes, and various other small RNAs. Now, so this is the subunits which we were discussing. We discussed about the transcription factors. So this tabular form, it shows us what are the different subunits and what is the role of these transcription factors. So as you can see, as I mentioned, so the particular order in which they bind, that is, so first and foremost, we have the factor D which binds. So obviously it has to have the function that it will recognize the data box, and it basically helps us, it regulates the binding by the TBPs. Similarly, followed by binding of the factor B, then F, E, and H. Again, the important thing about H is that it helps in the unwinding, it helps in the melting of the at the transcription start point. Also, it phosphorylates the serine 5 of the RNA polymerase C-terminal domain, and it releases the RNA polymerase from the promoter. As I said before, it helps in the promoter clearance. So, this was about the transcription in both the prokaryotes and the eukaryotes. I hope you have understood and the concepts are clear.